Greetings. My goal today is to talk to you a little bit about alveolates to remind you who they are and dig into the dinoflagellates and the apicomplexans. And then we'll go from there to ciliates in another lecture. So I will share my screen. So just to remind you that the alveolates are the A in SAR. Within the alveolates are the Apicomplexa, the who I'll talk about today, dinoflagellates and ciliates. They do have a synapomorphy, which are the alveolar sacs, but really they first emerged as a group based on molecular phylogeny, and the alveolar sacs were identified second. Here's a tree of all eukaryotes. Here's the group SAR we've been talking about, and alveolates are the A in SAR, with the dinoflagellates and apes, what I'm focusing on today. So who are the dinoflagellates? They're beautiful. They tend to have a flagellum that goes around their body. So dino for dinoflagellate means whirling, because they tend to spin, and then they'll have a trailing flagellum. These are SEMs. Here's another one where the groove for the flagellum, and another one. And this is a cyst, and I'll return to the cysts in a little bit. What are the features of dinoflagellates? Dinoflagellates can have armored plates, or be called thecate. Some have eye spots. Many are involved in symbioses. I'll say a bit about this in a little bit. They have strange nuclei called the dinokaryon. They lack nucleosomes. Some lack traditional histones. Some have their DNA in a liquid crystal form. We don't really know much about that. They have closed mitosis, and this is the clade where they have those crazy tunnels that I showed you in an earlier lecture. Plastid history, very complex, and I'll say a few more words about that. So here's just some line drawings of dinoflagellates. They have all of this special terminology that is unique to them, which I won't ask you to know, except to say that things like the location of plates and sutures is how taxonomy in dinoflagellates originally was done before molecular data. They have a rich fossil record, perhaps Precambrian. This is the cyst story. By 400 million years ago, there's some clear fossils, and lots at about 245 million years and more recent. Here are these cysts. So here's a recent cyst, 65 million year old, 100 million year old. And the reason why we know these are cysts of living ones is if you take a living armored thecate dinoflagellate and dry it out, you can see it transition from this form into this form, and you put the conditions right and it goes back. When we look at the evolutionary history of these forms, you can see here the width is the number of species. We're looking at 250 million years to the present. You can see, for example, this clade is very low number of species and then explodes and has maintained its diversity through today. Other clades like this one went extinct when the dinosaurs, the bulk of the dinosaurs went extinct. Most of these guys went extinct. These guys are doing just fine. So there's a rich fossil history here. As I said, plastids are really complicated. Molecular phylogeny shows several losses, many gains. The plastid genomes are really weird, small mini circle with single genes on them. And it all leads to this question of whether alveolates are primitively photosynthetic or whether photosynthesis is derived within. Some of you might read the paper with this figure in it. It suggests a photosynthetic alveolate ancestor with a loss into ciliates. We'll talk about them in the next lecture. We could come to apicomplexans in a bit. And if this, under this model, at this moment, at the base of all dinoflagellates, they gained peridinin, lost chlorophyll C, weird genomes, weird rubisco. We know that there was a haptophyte replacement in dinos, a diatom replacement, and really complex history within here. I'm going to add just a little bit more detail. Here's a phylogeny of dinoflagellates. These down to Noctiluca are all dinoflagellates. These are all photosynthetic, but the cryptomonad, this Dinophysis has a plastid, but it's clearly kleptoplastidy. It stole it from a cryptomonad. And both Amoeba phyra, which is a parasite, and Noctiluca, which is one of these blooming ones I'll talk about in a minute, no evidence of plastid. Oxyrus is sister to dinoflagellates and may have a little bit of past history of plastids there. So I leave it to you, plastid ancestral or not. If we go on from there to talk about their life histories, incredibly diverse. They're predominantly haploid, many forms of parasitism, many forms of endosymbiosis. If you like fish, perhaps you've heard of velvet disease, where your fish gets a velvet gleam to it, and that velvet gleam is because of these dinoflagellate parasites in the skin of the fish that are essentially feeding off of the fish. Not nice for the fish. Here's another parasite in a copepod. Here's the parasite sitting within, that's the dinoflagellate. Here's the copepod again with a living dinoflagellate inside. It poops out little dinoflagellates. But we have to be careful. Oh, and here's just a fluorescent image of these as well. They autofluoresce. This one I think has plastid still. Well, I'm not sure, but we have to always ask, because people always assume everything's a parasite. So we're going to look at the impact, and we're going to look at the survival of the copepod over time, whether they're infected or not. And so the dashed line are uninfected copepods and their survival, and the solid lines are the infected. So we can definitely see a substantial change of infected versus uninfected copepods, a real true fitness effects. But those copepods do okay and still survive up to 12 days instead of 18 days or 17 days with that huge parasite. Many dinoflagellates are involved in symbiosis. Coral reefs exist because of their association with dinoflagellates. So this is an animal in the Cnidaria. And here's the animal wall, and inside the animal are these dinoflagellates. Here's their crazy nuclei with these weird chromatin. And here's the plastid with all of its little thylakoid membranes. Here's the animal's polyps, and each little green dot in here is a plastid. So we have to thank dinoflagellates for coral reef, where they're called zooxanthellae. There are other animals that are green as well. A big fat green-lipped clam, a green-lipped sea slug, and we're going to watch a 2-minute video on these green sea slugs so you can hear somebody else's voice for a couple minutes and learn something more about them.
The world is thinking.
This is the sap-sucking sea slug tail, or the solar-powered nudibranch, or sea slug tail. So this is actually about a sacoglosson. Remember I told you about the different groups. This is not a true nudibranch. This is a sap-sucking sea slug. I'm going to talk about a group called Alveolates. Right? So remember that the sacoglossons have these piercing teeth. So here are some of the teeth here. And they use these teeth to pierce algae that are kind of like, that don't have cell walls. It's just a bunch of chloroplasts inside this. Once they pierce it, just chloroplasts and other cellular parts. So they pierce this, they suck up all of the insides, and they're what's called a kleptoplast. And that means they steal the chloroplasts from the algae. So we've had this like running theme of stealing from your prey, right? So these guys, Alveolates, they actually take the organelles, they take the chloroplasts from their algal prey and put them in their body, in their digestive tissue. So this Alveolates, the specific species that most of the work has been done on, feeds on a specific species of algae. It takes up and sequesters or holds onto its prey's chloroplasts. But the amazing thing is, not only does it take the chloroplasts continue to photosynthesize in the slug, which might be, okay, maybe it could photosynthesize for a while, but these can continue to photosynthesize because somehow, well, they continue to photosynthesize for long periods of time. And we know from the biology of the algae that the chloroplast needs proteins that the algal nucleus makes, not the slug. So 90% of its needed proteins are made by the algal nucleus, the DNA in the algae. So these chloroplasts are in the slug, but they don't have all the genes they need to function, to photosynthesize properly. So recently it's been found that the slug genome has, the nuclear gene that facilitates photosynthesis is found in the slug genome. And it's the same as what's found in the algal genome. So somehow elements of the algal genome have been transferred to the slug.
So pretty crazy. I encourage you to Google these green animals. There's many kinds, and people are beginning to understand more about the physiology. The lateral gene transfer is controversial, but still intriguing. So dinoflagellates can be friendly. They can also be harmful and form blooms, but not every bloom is harmful. So I'm just going to give you an overview of dinoflagellate blooms. I'm going to start with this one, Noctiluca, which I said doesn't have a plastid. But it is bioluminescent, and this is a boat coming out of a Noctiluca bloom. In this case, the dinoflagellate, I suppose it can be harmful just by its densities, but it's not producing any nasties the way the next few examples are. There are many toxic blooms, though. A nutritious meal of mussels can cause illness and even death when algal toxins are present. So probably many or most of you have heard of red shellfish poisoning. What I want you to know is there's many kinds of dinoflagellates that have evolved to have toxins, not to hurt humans, but it's a side effect. So there's paralytic, diuretic, neurotoxic, amnesic, and ciguatera. Here's an outdated map, but still shows us where you'd find neurotoxic in purple, and I'll talk about Pfiesteria at the end, amnesic in light blue. So you can see these are significant events, and many of you who come from coastal areas have probably heard about them. They also kill plants and animals. So these are the number of fish killing events in a 10-year period up to the year 2000. These have continued on. They can, in fact, kill whales. That's very sad, so we won't look at that very long. Instead, I'll just give you a few tastes of this. Here's a genus that causes neurotoxic blooms off the coast of Florida. This has been in the news in the past 5 years. It has a global climate change link to it. And here's a history of some of these events in the Gulf and along Florida. Alexandrium is a genus that causes paralytic shellfish poisoning. And here is where it's found in the north and the west coast. Ciguatoxin is a reef fish, and people in Puerto Rico are well aware of the danger of this dinoflagellate. It causes neurological problems as well. So it's in Florida and Puerto Rico and Hawaii, tropical waters. And you can also have these diuretic ones. This one, I believe, is fresh water. So all of that color is the dinoflagellates. Not all red tides are dinoflagellates. Here's a diatom called Pseudonychia, which causes an amnesia where you lose your memory. And here are where those events occur. I want to end the dinoflagellate story by just telling you the tale of this complexity of a dinoflagellate genus called Pfiesteria, which led to Pfiesteria hysteria. If you're from North Carolina or the Chesapeake Bay area, you might have known about or seen the reoccurrence of these fish kills. That's a beer can. These are all fish. And when people pulled up these fish, they had these holes drilled into them. This was the occurrence of what they suspected to be the causative agent. And here's Baltimore and Washington, D.C. in the Chesapeake Bay. The dinoflagellate Pfiesteria was named after Dr. Louis Pfister, who did show that dinoflagellates can have multiple life cycle stages, but in this case, the claim was 24 different stages, and the end of the story is not very convincing. But the claim was that this was the life cycle of the dinoflagellate in this marine setting. It could be an amoeba, it could be a toxic spore, it could be a non-toxic spore. It would eat the fish, kill the fish, become an amoeba, encyst, do all these crazy things. Life cycle of one organism. These were pictures from the studies that said this one dinoflagellate can do all these things. The problem was to get the dinoflagellate to do its thing, you have to add a fish, and you can't add a sterile fish. So the alternative is that some of these amoeba, for example, here's lugalls, we use that in lab. Some of these amoeba came in with a fish. Here's just some more pictures of these. There's one with a beautiful flagella, another one. Now this is an amoeba. Could it be a dinoflagellate? I don't know. So in the 1991, 1997, there were these huge kills. 1 billion fish were killed. But more importantly, at this time, these are facts. There were effects that were linked to these explosions of these fish kills: skin lesions, red eye, headache, nausea, vomiting, short-term memory loss, and in some cases, for the scientists who work with us, severe cognitive impairment, disorientation, speech. They became a biohazard 3, this organism. And there's a book you can read called "And the Waters Turn to Blood," telling the story of this organism. Lots of controversy here, but in the end, the results weren't repeatable, so the whole thing blew up. Very controversial. You can still follow the debates. What is a fact are the effects, and the fact where the fish kills, whether it was this organism or not, I don't know. And then the other angle were the politics, because it was farms like pig farms upstream putting pollutants in the water that were argued to be associated. So the punchline here then is that there's a dinoflagellate suspected of being a major killer. It had big political ramifications as well as the scientific ones. So I'll spend hopefully maybe a little bit less time on the Apicomplexa. They are crazy organisms. They're obligate parasites. You're going to read about them. They have infective spores, so they were called sporozoa for many years. They all have an apical complex, apicomplexan, and it consists of these words that I don't really care for you to know. They have a reduced plastid, many, but not all. They have this semi-open pluromitosis that I taught you. This is the apical complex. Basically, I see it as a drill bit. These are microtubules, and this is like a little drill bit. And below the drill bit, this is the front end, the apical complex, you have these rhopteries, these bags of nasty enzymes. So combining the drill bit and the nasty enzymes, this is how these organisms penetrate their host or their host cells if they're intercellular. So here's just another picture. Here's the nucleus. Here's crazy mitochondrion in these guys. And here is a apicomplexan drilling in. There's the drill bit and the bags of chemicals. And this would be host cell, apicomplexan. Historically, they were broken down into primitive gregarines that could be intra or extracellular, a group called the coccidia, which included toxoplasma that I'll tell you about, and cryptosporidium, which I won't, which causes diarrhea. And then the hematosea, the ones that live in blood, these are all intracellular, and malaria is in here. They have an obligate alternation of hosts. So this was what the phylogeny was thought to be, primitive to complex. Of course, you guys see that that probably isn't true. Maybe gregarines are everywhere. You're going to read a gregarine paper and come back to it. So historical molecular data. I'll tell you just about two, the genus Toxoplasma. Apparently, this is the one that women are supposed to wash their hands when they're pregnant if they have cats, because it can pass the blood-brain barrier of developing fetuses. It's very dangerous. Some people claim it actually comes more often from undercooked meats. So it's its transplacental role in developing babies that's most important. Some people estimate that 50% of Americans, for example, have been infected, and normally we have no symptoms. I think many of us who grew up with cats may be infected. Maybe every once in a while they come out in our brains and spin off and we say, oh, I have the flu today, and the next day our immune system suppresses them. So Toxoplasma, its final host is a cat. Here, this is where the toxo goes through its sexual cycles, but it can also pass through mice and it can pass through humans, but ultimately that needs a cat to do the sexual cycle. So, excuse me, we call it the final host. But very scary, Toxoplasma in patients with AIDS, you can get a reactivation of an older infection. So you don't even need to be reinfected if you're sadly patients with AIDS. This often occurs in the brain. There are toxo that affect the eyes. You end up with fever, headache, confusion, and seizures. And you can treat these, but you cannot eliminate this parasite. And here is a brain lesion. These are brain lesions in an AIDS patient where you've had a reactivation. So in places where HIV isn't able to be maintained in a suppressed level, this can be a really big deal. I'll end with Plasmodium, which is the cause of malaria is in this clade. Its final host is mosquitoes, but can also pass through many vertebrates, and there are many species of Plasmodium out there. This is Vivax, which can go through a mosquito, where it goes through all of its sexual life cycle stages with all of these different words, which I'm not gonna hold you to. It can get into the human blood cells here, and I told you guys about how it can, a single blood cell can fill with nuclei, which then splice their way out and go round and around in the human life cycle. And I showed you already this picture of red blood cells with the malaria peeking out. Here is red blood cell with malaria, uninfected red blood cell, infected red blood cell, and here are the little forms, in this case called sporozoites, bursting forth. I told you already that in malaria, we have this semi-pluromitosis. You have multiple mitoses. It's semi-open, little holes, comes in from an angle instead of straight on. If you have malaria, sadly, you have fever, amnesia, obstructed blood vessels. There are some treatments, but there are many drug-resistant strains, and malaria remains a tremendous problem in many parts of the world. One of the things these crafty critters do is, this would be a malarial chromosome. They have sort of traditional genes in the middle, but subtelomerically, they put their genes involved in immune resistance. In this case, they're called VAR and RIF. They sit here at the ends, they evolve really rapidly, which is how they escape our immunity. And all of the 14 malarial chromosomes end with these genes that are evolving rapidly. And even more interestingly, not only do the genes end with these chromosomes, but it turns out that only one of them is expressed at any time, and that depends on where it is in the nucleus. So basically, you can have many different, it's like somebody has taken your wallet and is running away, but they're changing their clothes. So this is a blue jacket, a green jacket, an orange jacket, and they just keep switching around so that our immune system can't recognize them. So the last thing I'll say about malaria, Plasmodium, is that they have something called the apicoplast. They have this strange little organelle in here, no thylakoid membranes or anything. But when you look at this, it is clearly a remnant chloroplast. It's not in every apicomplexan, but it is in the toxoplasma and the Plasmodium. It still has a genome that codes for 30 proteins involved in DNA repair, replication, and transcription, but other metabolic roles like amino acid and fatty acid biosynthesis and starch storage. So people have been very interested in the apicoplast, the plastid in the apicomplexa, as a target for drug design. So I hope that that gives you a sense of the alveolates in general and the dinoflagellates and the apicomplexans in specific. Take care all.